This screencast will deal with how to read results on a Lancefield grouping. Lancefield groupings are used to group different kinds of streptococci so we can identify the different groups. We run a series of biochemical tests to help differentiate the group of streptococci. When we look at a Lancefield grouping, there are a few different tests we can deal with. Number one is how does it grow on blood agar? The sample is streaked or is inoculated across a plate of agar that contains 5% sheep erythrocytes or sheep red blood cells. The organism may produce hemolysis, which have the ability to rupture the erythrocytes or to destroy the red blood cells and produce a clearing on the blood agar. The way to observe this effect is to illuminate the plate from behind. Then look at where the colony and the bacteria are growing and observe the edge. It is along the edge that changes can be observed. With beta hemolysis, there is a distinct zone of clearing around the colony. The agar will appear transparent. You can see through it. Anything on the background will be able to be seen. This is a result of complete hemolysis or beta hemolysis of the red blood cells in the agar. The second kind of hemolysis is partial hemolysis that is called alpha hemolysis. With this, there is often a faint greenish color around the colony. The agar also gets a green tint to it, referred to as a greenish discoloration. This comes from the partial lysis of the red blood cells, without full breakdown of their contents. The last form of hemolysis is what we call gamma hemolysis, or no hemolysis. On this, the red blood cells are not broken down, are not lysed, but the bacterium still grows on it. Besides reading hemolysis on blood agar, there are also 3 antibiotic discs, the SXT, Optocin, and bacitracin, that are placed on the blood agar. If the antibiotic inhibits the growth of the bacterium, as it's happening with this particular antibiotic, then that is a positive reaction. If there is no inhibition of the growth, as it's seen on this particular antibiotic, then the reaction is negative. Another reaction that occurs on blood agar is the Kemp test. In the Kemp test, a line of Staphylococcus aureus is streaked right in the middle of the blood agar. Then, a line of the Streptococcus sample being tested is streaked perpendicular to the Staph aureus up to but not touching the line of Staph. Both the Staph and the Strep will release a hemolysin, but one group of Strep releases a hemolysin that will react in a synergistic manner with the hemolysin of the Staph, causing a larger clearing where the sample meets the Staph. In this case, the synergism will show as a large arrowhead at the end of the Strep sample. The formation of this arrowhead is a positive Kemp test. The first of the other 2 tests we have to perform are growth on 6.5% NaCl broth. The broth has about twice the salinity of seawater, making it a hypertonic solution. Most organisms cannot grow in this high concentration of salt. Growth is demonstrated by a cloudiness or turbidity to the medium, which is a positive result. This means positive for salt tolerance. No growth looks like this one, shows a clear broth, a see-through, is a negative result. The last test we'll talk about is the Bioskalene. Besides other ingredients, this medium has bile and iron. With the decomposition of the esculin, there is a precipitation of the iron, giving the medium this dark brown to a black coloration. The positive reaction is therefore a dark brown to a black coloration, and the negative reaction is a translucent light brown, like a tea color. If we take these tests and place them all together, then we can begin to identify the group to which the specimen belongs. In this particular case, if we look at this unknown number 1, we can see it is Bioskalene positive, which means that we would look in the column of the Bioskalene. It says it is going to be either group D enterococci or a group D non-enterococci. We can see that this particular one is NaCl positive, salt tolerance positive. If you observe the table for Streptococcal ID, you can see that NaCl positive means that it could be the group B, Streptococcus agalacti, or it could be the group D enterococci. So you will see the only one that has the two of these tests, bioskalene and salt tolerance, as a positive is group D enterococci. As you keep looking on the line for the group D enterococci, you see that it should be negative for SXT, optocin, and bacitracin, which is confirmed by the nuclear zone around the antibiotic disc. And as you continue to look at the line for the group D enterococci, you can observe that it could have either alpha, beta, or gamma hemolysis, and it would be negative for the Kemp test. So that's what we have up here. So when we read these Lancefield groupings, that is the way that we would do it. Please note that most of these tests will be negative. So start with the positive tests for each one of the unknowns you have to complete. Also, the upper part of the table shows beta hemolysis and the lower half does not. So once you learn what each test does, this process of identification is going to be rather simple.